Hello dear science enthusiasts. Today, we will explore an incredible topic that pushes the boundaries of science and technology, artificial DNA. Could this technology truly possess the power to recreate life? While delving into this question, we will also examine the extraordinary potential of artificial DNA and the frightening risks it entails. Imagine being able to rearrange human DNA to make us completely resistant to diseases, enhancing our physical and mental abilities to incredible levels. We could genetically reshape ourselves and become healthier, stronger individuals. However, alongside these promising opportunities lie significant and dark dangers. Artificial DNA could be used to produce biological weapons, create new and deadly viruses, or develop entities that threaten humanity. Managing this technology correctly in terms of ethics and safety is crucial for the future of humankind. So, let's start exploring this exciting yet terrifying topic together. DNA stands for deoxyribonucleic acid and forms the genetic material of all living things. DNA carries the information that determines how an organism looks, functions, and develops. This information is stored in sequences of nucleotides made up of 4 bases: adenine, thymine, cytosine, and guanine. You can think of DNA as letters on the pages of a book. These letters are arranged in a specific order to determine all the characteristics of the organism. Now, let's take a look at artificial DNA. Artificial DNA is a laboratory-created copy of natural DNA. This process, which began in the 1970s with the synthesis of the basic building blocks of DNA, gained significant momentum in the 21st century with the discovery of CRISPR technology. CRISPR is like a pair of scissors that can cut and alter genetic material. This technology plays a crucial role in the creation of artificial DNA. Artificial DNA is produced in a laboratory environment through several steps. You can think of this process as rewriting the pages of a book. Natural DNA is a book we are born with. Artificial DNA is rewriting the pages of this book in a lab. First, we start with the synthesis step. The nucleotide building blocks of DNA are chemically combined in a specific order. This step is like assembling the letters of a book one by one. Then, in the amplification phase called PCR, polymerase chain reaction, the DNA sequence is replicated. This step is similar to making copies of a book. Next, we move to the cloning phase. The produced genetic material is usually placed into a vector such as a plasmid. Plasmids are small circular structures used to transport DNA. This step is akin to converting the written book into a portable format. Finally, in the editing step, the DNA sequence is altered as desired using gene editing tools like CRISPR/Cas9. This step is like changing and editing specific parts of the book. After learning these steps, let's look at the treatment opportunities offered by artificial DNA. Artificial DNA can provide groundbreaking solutions in the treatment of genetic diseases. Genetic diseases result from faulty genetic information in DNA, causing certain genes to work incorrectly or not at all. By correcting this faulty genetic information with artificial DNA, diseases can be treated. For instance, it might become possible to treat hereditary diseases like cystic fibrosis at the genetic level. Cystic fibrosis is a disease that affects the mucus-producing cells in the body, causing severe problems in the respiratory and digestive systems. Treating this disease genetically can significantly improve patients' quality of life and potentially eradicate the disease entirely. Think about its potential in cancer treatment. Artificial DNA has the potential to revolutionize cancer treatment. Cancer is characterized by the uncontrolled proliferation of cells, often caused by genetic mutations. With artificial DNA technology, the genetic structure of cancer cells can be targeted for treatment. For example, using CRISPR technology, mutations in cancer cells can be corrected or these cells can be destroyed with specific gene-targeting methods. This allows for the development of more precise and effective methods in cancer treatment. How about its impact in the field of agriculture? Artificial DNA can be used to develop more efficient and resilient plant species in agriculture. This can significantly contribute to global food security by increasing agricultural production worldwide. drought is a serious threat to agricultural production globally. Artificial DNA technology enables the development of drought-resistant plant species. These plants can provide high yields in areas with limited water resources, enhancing agricultural sustainability. For example, genetic modifications with artificial DNA can create plants with high water retention capacity, making them more resistant to water scarcity. Another significant issue in agriculture is plant diseases. These diseases reduce agricultural yields and cause severe economic losses. Artificial DNA technology can be used to make plants resistant to diseases. Let's look at examples of developing resistance to plant diseases. Using artificial DNA, genetic modifications can strengthen plants' natural defense mechanisms. This makes plants more resistant to diseases and increases agricultural production. The role of artificial DNA in environmental sciences is also crucial. Artificial DNA can play a significant role in solving environmental problems. Solutions such as bacteria that can clean pollutants or microorganisms that can produce biofuels are vital for sustainability. Pollutants released into the environment from industrial activities and accidents pose a major threat to ecosystems. Artificial DNA technology enables the development of bacteria that can clean these pollutants. For example, bacteria that can clean oil spills can biologically clean oil pollution in seas, protecting ecosystems. These bacteria are genetically modified using artificial DNA to break down and clean pollutants more effectively. The use of fossil fuels contributes to global warming and environmental pollution. Artificial DNA can be used to develop microorganisms that produce biofuels. These microorganisms convert organic waste into biofuels, creating renewable energy sources. For instance, genetically modified algae with artificial DNA can be used to produce biofuels through photosynthesis. This reduces dependence on fossil fuels and offers eco-friendly energy solutions. Alongside all these potential benefits, the risks and ethical dimensions of artificial DNA technology must not be overlooked. Artificial DNA technology has the potential to revolutionize many fields such as health, agriculture, and environmental sciences. It can be applied across a wide spectrum, from the treatment of genetic diseases to increasing agricultural efficiency and solving environmental problems. However, it must be handled carefully in terms of ethics and safety. Interventions in human genetics can affect the genetic makeup of future generations, leading to serious ethical debates. There are deep ethical questions about the boundaries and possible consequences of genetic interventions. Artificial DNA technology, when misused, also carries dangers such as the development of biological weapons. Strict regulations are required to ensure the safe use of this technology and prevent its misuse. Let's consider examples of these risks. For instance, dangerous viruses or bacteria developed using artificial DNA could cause massive human losses. Dr. Jennifer Doudna, one of the pioneers of CRISPR technology, makes significant evaluations about the potential and risks of artificial DNA. According to Dr. Doudna, genetic editing technologies can be a great source of hope for humanity, but they also carry great danger. The misuse of these technologies can lead to the development of biological weapons or increased genetic inequalities. In conclusion, while eagerly anticipating future developments in artificial DNA, we must remember that this technology needs to be used responsibly and ethically. New research and technological advancements will continue to expand the applications of artificial DNA. Studies in this field will continue to push the boundaries of genetic engineering. The impacts of artificial DNA technology on society should also not be ignored. Issues such as access to genetic information, legal regulations of genetic interventions, and the societal acceptance of these technologies open up a wide area for discussion. Legal frameworks should ensure the application of genetic engineering within ethical rules. Educating and informing society about artificial DNA technology is crucial. Educational programs and awareness campaigns should be organized to ensure proper understanding and ethical use of these technologies. In this article, we have provided detailed information about the potential and dangers of artificial DNA technology. This technology, with the potential to revolutionize many fields such as health, agriculture, and environmental sciences, also brings significant ethical and safety concerns. Which aspect of artificial DNA do you find most intriguing? Share your thoughts with us in the comments. If you want to join this exciting world and learn more, don't forget to subscribe to our channel. If you liked our video, don't forget to hit the like button